Yeah. Can you speak? So your slide is okay. So I just want to hear your voice.
Okay. Hello
Hello,
everyone.
everyone. Yes, we can hear you. So please start.
Okay, thank you for still here. Okay, it's already quite late evening here in Malaysia, I think. And good afternoon, good evening to everyone. Thank you for the organizer for having for organize this event. And sorry, I have to turn off my video because we have a very poor connection since we have a very heavy rain since this morning. So I have to turn it off so that I can get a better uh, connections. Okay. And today, on behalf of my team, I would like to share some of our findings when we are using a few analytical techniques on analyzing and determining the microplastics in biota sample that we have done recently. This work is actually a collaborative work that we did with our partners, ALS Technical Malaysia, where they are the the lab that doing the pyrolysis for us. Um, and yeah, hope you can stay with me. Okay. Before we go into any more details, let's zoom out to the current situation of marine plastic debris and microplastic pollution in global scale. And I think everyone here is already familiar, very familiar with this data from Professor Jana Jambek that has been published in 2015. Where she highlighted the input of this pollution are largely coming from the Asia and the Southeast Asia regions. Okay, this data is from 2015, but I think even with the recent numbers change a bit. But however, this region is still top countries that need to work hard to manage this issue. That's my personal opinion. And when we zoom into the Southeast Asia region, okay, from the last 10 years, we found that from the Scopus Web of Science, Google Scholars, that more than 150 or more than 119 articles that has been published for the keywords microplastics and marine litter or mesoplastic that has large size than 5 mm but still in less size than 25 micron sorry. However, more than half of the articles, especially for the mesoplastic or large microplastics, only characterization based on physical analysis. There are few problems because of underestimation or overestimation of the real data. And then if you can see from the recent scenario since 2019, Indonesia and Thailand are currently gaining a very big momentum in this area. I think because they have a very good support from the institution in the government sectors in both countries. Uh, but if we're talking about Malaysia, because I'm coming from Malaysia and I think I'm the only Malaysian that been able to present in the symposium in 3 days. If you look into Malaysia context, since 2012, 10 years ago, we published more than 40 articles, and we note that more recent years that instrumentation analysis has been used regularly to characterize the microplastic. However, if we look deeper on this data, we are still using ATR, FTIR that still will underestimate or overestimates the amount of microparticle or the type of polymers that's associated with the microplastics or nanoplastics. Because is for larger particle, right? So I think this problem is because of the availability, and other than that, is a types that the cost, the issues about the fundings and so on in Malaysia. Okay, so we know that biota such as a deposit feeder or filter feeders, okay, such as bivalve, mollusks, polychaetes, um, and fish can accumulate the microplastic through their feeding habits and activities. Okay, so this will lead to the transfer of these contaminants into food products and our food chains, and would trigger health risk issue later on. And there's a lot of challenge in identifying, determination, and characterization of these type of contaminants, especially in a small size of sample of this type of biotas. That has been highlighted by many publications and researchers recently. So, in our approach, that we know that traditional quantitative method alone, okay, such as you just do the physical sorting alone, is not enough. Okay, it's not enough for without the support of chemical analysis to be able to identify and analyze the polymer associated to the microplastic. So you won't really know that is it organics or inorganics or polymers and etc. So in our case, we opt to use the thermal degradation method of pyrolysis GCMS, and also we also use the micro FTIR analysis in order to be able to identify the type of microplastic in two different biota metrics, polychaete and fish tissues. Okay, the samples, all the samples were collected around the estuaries and the fish markets at the South China Sea, it's the east coast of Malaysia, where all the samples are being oxidized, being extracted and digested using a wet peroxidation method, following the international guidelines, UNEP. Okay, so so this is the overall what we say, the overall or the general flow of our analysis. So we have a polychaetes, and also we have a fish from the 4 species. Polychaetes is we have it for 40 samples that we pull into 10 each for the digestions, and the fish collections also for the 4 species we also have each of them is 10, and then all these being extracted and digested based on physical sortings, and also after that chemical characterization techniques. Okay, so okay, so further, if you go into the detail for chemical characterization, the chemical vibration spectra and profile for microplastic samples were analyzed using the FTIR microscopy. In our case, for this purpose of presentation, we present the one that we use with Lumos micro FTIR. Before the analysis, the filter paper containing microplastic placed directly onto the sample holder, and we use the ATR germanium crystal to press against the samples, and we do it point-to-point analysis for this because we are using ATR. And then this is really not recommended if you have a very small size of samples, as mentioned earlier by a few of the keynote speakers. But using this technique, we able to point our microplastics that the analyte of interest that we want to analyze. And then we set the matching, and then all the data being interpreted by open software and library. And then for the pyrolysis that has been done by our collaborators, This technique utilizes the high temperatures pyrolyzer. In our case, is 600 degrees Celsius to pyrolyze all the microplastics and maybe the additives before the compound was separated in the GC, and later on the mass ion is analyzed by the MS. So in this case, we are using the non-polar HP5 MS column. And then, before that, before the analysis, prior to the analysis, the sample was cleaned up and treated with tetramethyl ammonium hydroxide or TIMA reagent. This is to promote the thermocatalysis reaction for the sample. And then this is the analytical parameters for the pyrolysis that being used. Program that being used. The Pyrolaser, as I mentioned earlier, we use 600 degrees C for pyrolysis temperature. And this pyrolyzer is attached to the GC that coupled with the MS. And for this program, the pyrolysis period is 10 minutes. And the transfer line was heated, and we maintained the transfer line for 320 degrees, I think. And in the GC, the initial oven temperature was set at 40 degrees before it will ramp up to 300 degrees C at 20 degrees per minute before the final temperature was held at 10 minutes. So it's a very fast analysis process. We use it for the inlet. It's a speed mode, 95:1, with the flow rate of 1 microliter per minute. And for the mass spectrometry, the analysis will run under a selective ion monitoring mode. This is the selecting ion monitoring ions for the selected polymer. We have 8 polymers that we would like to see from this analysis. When we already pre-select these selective compounds to monitor their fragment ions and the monomers for each polymer types. So we have a PA6 is polyamide 6, nylon, polyethylene, polyesters or polyethylene terephthalates, PMMA, polypropylene, PS, PVC, and polycarbonate. These all 8 polymers are very common in our environment. So this is when you are dealing with microplastics analysis, the most important thing that you have to consider, I think all the steps is very important, but one of the most important things is the contamination control consideration, since we know that there are also airborne microplastics, since nowadays people already publish a lot of airborne microplastics, so we have to minimize that. So these are some contamination control that we have to consider. This is a very general one, but in our lab we have a clean sorting room that we dedicate for microplastic extraction and physical sorting analysis. So in this room, everything is being really cross contaminated to avoid or to minimize the airborne microplastics. So so for the results, a total of 314 microplastic extracts from polychaetes, and dominated by straight fiber-like microplastics and transparent and brown colors. So from this can be derived from the decomposition of synthetic fishing gears, because the major activities around the estuarine areas are agriculture or fishery-based activities. So this might become the provider to the microplastic pollution to this area. And the domination of the transparent colors and brown colors are well expected. As I can say, it's probably due to the decolorization process, maybe because of the aging photodegradation process of the microplastics. And we know that the diets of the polychaetes, because this is for polychaete samples, polychaete swarms are dependent to the food source from their habitats. In our case, these polychaetes found in the nipa tree. So the nipa, so they eat the nipa tree, and then they might be mistook the microplastic as their food, and accidentally consume microplastics as their diet. So this is the example of some microplastic particles that we found using the physical analysis. And for the fish tissues, a total of 411 microplastic can be retrieved from all species in the pool, mostly in also in a thread and fiber type, fiber type, and also black color. Note that this number is a bit lower than any other number that been published before, because for this particular study, we focus on the microplastic in the fish tissues, where we know that for fish, actually the microplastic usually found in the GIT, the stomach, the gastric content, but we would like to see if this small amount of microplastic in the tissue, edible tissue, because that's the thing that we consume here in Malaysia. So these are the example of microplastic image that we can capture using the physical analysis. So this is come from the chemical analysis. For the micro FTIR analysis, we found that 8 polymer types that can be identified using this technique. Okay, for this analysis, we only use a 5 to 10% of counted samples to characterize using this method. The 8 polymers that we're able to identify using micro IR are rayon, semi-synthetic plastics, PP, PE, PET, PVA, polyvinyl alcohol, PCs, polycarbonates, PVMK. PVMK is a polyvinyl methyl and nylon. And among the semi-synthetic polymer of rayons and PP dominated the identified polymers. So this has been characterized and matched from the polymer library. And if we can see that this is the example of micro FTIR spectrum and chemical image that taken with this technique. We have a chemical mapping or imaging here, and also we have a spectrum that we can produce from the system. So the background spectra actually were collected once before every samples, because we are using ATR, so we have to make sure that we minimize the contamination, because we press the crystal against the samples. Okay, so actually this technique is very suitable for identification of irregular shape of microplastic particles, since we are doing point and shoot. Most of the time, microplastics consist of many copolymers that possibly having very different monomers and functional groups. So this may also lead to the complex IR spectrum and interpretation. So I think it's very good that we are able to match the profile with the library, but we also need to have some basic chemistry to identify the polymer properly. That's my personal thoughts. Okay, so when we analyze the sample with other techniques, pyrolysis, GCMS, this figure shows the example of total ion chromatogram, or what we call TIC, for selected sample based on targeted SIM mode that we choose before, of polymer decomposition compounds. And this table summarize the concentration data in microgram unit per liter actually. So it's a ppb unit from this analysis. So this table also indicated that the calculated microplastic concentration was based on individual calibration curve for the standard polymer before we run for the real sample. Okay, So this is based off from the 8 individual standard polymers. So interestingly, using this method, polymers such as PP, PE, PET, PVC were found in most of the sample, and some of other polymer also were found. However, in the trace level, you can see show peak here, but we cannot count the concentrations. But when I think we're still in optimizing this method as well. We can say that the accuracy and practicality of this technique is very suitable, especially when we consider we have a very analytical amount of sample, trace amount of contaminants and their additives. So I think this technique is very suitable. So because you mind that this coming from the flesh tissue, okay? When microplastic says to hardly able to penetrate the flesh tissue, it's not in a large amount as much as in GIT. So, but still we can manage to find some in our samples using the GC pyrolysis, GCMS. So I think it's a very good techniques. So after all, we know that these South China Sea regions are heavily affected by wind, by ocean currents, and such as monsoons. Now we are having monsoons in Terengganu, and this will be giving a very interesting findings in biota sample during each season. So that's why we would like to know whether the increase of the rainfall on the northeast monsoon or the southwest monsoon will bring more of this pollutant from land to the rivers, and finally maybe accumulated into the mangrove, into the estuarine, or into the ocean, that will affect all the marine life in this ecosystem. So I think there are still rooms that we can explore for this for this study. And then before I stop, here's some of the other challenges, especially in Malaysia context, since we still need more people and more research to be done in this area. Because what I think is, at the end of the day, whatever goes into the rivers and our ocean will come back to us and harm us. So this is some example from our findings that we found similar microplastic samples that we found earlier in those biotas, also found in the human tissues. So this is a very interesting topic, because this poses a health risk concern for this issue. So I think because our preliminary studies also prove that there's microplastics in the human community that likely originated from the environmental contamination. And to sum up, here's our group. We also monitor microplastics in other matrices, not only in biota. We also publish papers in sediments and also waters, fresh waters, and also oceans, marine. And we also collaborate with other researchers to find solution and alternative for this issue, that trying to be able to address all the UN sustainable goal. So I don't want to talk to take your time. Okay, I would like to acknowledge, and I'm very humble that and thankful for our collaborators, in which without them we cannot do this alone. We also welcome all the potentials new collaborators, especially for more works on microplastics in Malaysia, South China Seas, and Asia Pacific region. So we are welcome everyone, and thank you. I think that's very short from me.
Thank you, Dr. Sabiqah, Tuan Anuar. So it was a wonderful